Hello and welcome to today's Applied Medical Sciences taster session. And this taster session is titled Revenge of the Bugs and How Our Body Strikes Back. Thank you all to those who are joining us today. My name is Aryan Panchal and I'm a second year applied medical student here at UCL, and I shall be your chair this afternoon. So now, essentially, the whole point of this taster session is we're hoping that this will address, course-specific questions that you may have that maybe our prospectus doesn't really answer, or you might have any other question you want to ask about this course. And we're also hoping that this really gives you an insight of how it is like to actually study applied medical sciences, the undergraduate program at UCL. So this afternoon, we're very fortunate to have two special speakers. We have Professor Jennifer Ron and Dr. Andrew Williams that will introduce you to some of the great science and medicine that we will learn here as a student and what I've learned here so far in my course of applied medical sciences. So Professor Ron will first provide a summary of the applied medical sciences programs. And Professor Ron is the head of the Center for Urological Biology and runs a research laboratory studying treatment-resistant urinary tract infections. Professor Ron is based in UCL's Division of Medicine, and she is also a deputy director as well as the admissions tutor for applied medical sciences. So she knows and she sees a lot of applicants that come and apply to this course. So feel free to ask her about any tips or anything regarding the whole admissions process. She is also the program lead for the intercalated BSc in Clinical Sciences. And we also have Dr. Andrew Williams. Dr. Andrew Williams has a background in immunology and respiratory biology. He is based also in the UCL Division of Medicine and is actually the year 3 lead for the UCL MBBS medicine program, and which means he oversees a diverse portfolio of our BSc programs from UCL and external universities. So before I hand it over, this session will run for around 35 minutes, and thereafter we'll have a question and answer session where you guys can answer any question to our professors here and anything from applications or anything specific you want to know about the course. So now I'll hand it over to you, Professor Ron. And once again, thank you for joining us.
Thanks very much for the really nice introduction, and welcome all to this Taster Day. Really excited to tell you a bit about our course, Applied Medical Sciences, where our motto is fusing science with medicine, and that really says the whole thing. Our course is an in-depth look at how science and medicine interact, and it gives you a fluency in both of those disciplines. And we're really super excited by this course, and it's led by our program director, Professor Dave Spratt. Now, just before we do the taster bit and talk about the bugs, it's going to be a very, very brief tour of the course and a little bit of key information, just in case you're interested. So the Division of Medicine is the largest department in our Faculty of Medical Sciences, and it's really research intensive. So we have a mix of internationally famous clinical and non-clinical scientists all working together. It's about 40% clinical, 60% non-clinical research. We really have very close contacts with world-leading hospitals in London. As you probably know, London has amazing teaching hospitals, and we are very closely linked with those doing research and trials with them. We really do well at interdisciplinary research, teaching, and clinical practice, and we have a really high impact. And if you look at, I don't know if you care about the league tables, but the UK does a thing called the Research Excellence Framework, and UCL came in first in the UK for research power in medicine. And I mean, that's really exciting. So it's a high power place with a really great research. And our course, as I mentioned briefly before, we'd like to develop science graduates who have a strong medical background. So this is a science degree, but it's really clinically oriented. This will give you a strategic advantage if you're applying for any job that requires a deep knowledge of both science and clinical medicine. And it's a very personalized and tailored degree in that it's broad, but you can choose a narrow path, and we can get you where you want to be more specifically. And we do offer a lot of help in guiding you through the module so you can really pick your dream path to your dream job. And there's lots of different things you can do with this degree, and we'll talk about that in just a few minutes later. I want to highlight one thing about our course. So we get a lot of, first of all, we get a lot of students who wanted to go into medical school and didn't get in, and they come to us instead. And some of those students, we end up sending them out and they get into medicine after. So it's a very good place if you want to consolidate and you really want to be a doctor. It's a good place to go to get those skills, because we've had very good luck placing our students in graduate and regular medical programs around the country and around the world, actually. The opposite is true too. We have people who come in who, they want to be doctors and they end up really wanting to be scientists because they just, they loved biology at school and they thought they wanted to be doctors, but once they were exposed to all the amazing science out there, they changed their mind. And then there's people who come in and they're not really sure what they want to do at all. And I think this course is great for that because it's very broad, and it's hard to choose what you want to be when you graduate if you don't really know what the options. So we try to really help you find your dream topic, and also we give you lots of career guidance to help you get to where you want to be. So I think that's a unique point about our course. And people always want to know what happens to our students. We've been keeping close tabs, and our students are really highly successful, and some of them go straight into employment. Here's some examples here. They become clinical trials managers, they become consultants, not medical consultants, but sort of like business consultants. They work in public health, they work for the government. We've had somebody who's working for number 10 Downing Street. They work in finance, patent law, science communication, a whole range of interesting jobs that are allied to science. But most of our students go on to further study. So as I mentioned, medicine is a very popular destination, as is the PhD, and it's pretty much neck and neck every year. So with masters, so further studies in masters, and then medicine and PhD tied for second place, and then all these other jobs. So, and then it's really fun watching our students develop. But after they do the masters, many of them go on to do PhDs and get really interesting jobs. So we're quite successful at placing our students. They get a really good, solid background. Okay, now I'm going to do the taster bit. So to give you a flavor of the kind of teaching you might get if you come and join us on this course. And I'm a microbiologist and a cell biologist by training, and I'm really interested in bacteria. So I'm going to talk a bit about Revenge of the Bugs. And when I say bugs, I mean bacteria. And Dr. Williams is going to give you a little bit about immunology after that, which he's a bit more focused on viruses. And let's just dive in. So bacteria are really interesting because they were the first form of life on the planet. You probably know this. 3.5 billion years ago, just when the Earth had cooled up and had become amenable to life, life began. And this was ages before the first eukaryotic organisms came on the scene, 1.5 billion years ago. And the first mammals didn't come for 200 million years. So on this timeline, it's just a little pixel at the end when we showed up. So as you can imagine, the bacteria were here first. They were here for millennium before we got here. So obviously they're really clever. They're small, but they're clever, and they really know how to push our buttons and to get around our defenses. And I think and this picture is just some beautiful structures, which are ancient bacteria that have formed communities and mats, and they make these really beautiful rocks. And this is from Australia. So this is just some evidence that bacteria are very old. And you probably all heard of the Black Death. Killed probably about 50 to 60% of the population of Europe back in the day, back in the 1300s. And there's nothing you could do at that time if you were infected with the Black Death. You basically had to get the local priest to pray for you and sprinkle you with fairy dust. You're basically a goner. So bacteria have been devastating humans for as long as humans have been around. And what causes it? They're tiny little things. Black Death, the bubonic plague, is caused by a bug called Yersinia pestis, shown here in the gut of a flea. And the fleas live in the rats, and the rats obviously are around human settlements, and that's what spreads the bubonic plague. But they're only tiny, they're 2 microns. How can something that has no brain and is only 2 microns in width how can it devastate and kill half of Europe? It's a good question. And the reason is because bacteria have all these amazing ways to get around our defenses. Our defenses are formidable, right? We are highly evolved animals with amazing immune systems. And Dr. Williams is going to tell you more about that when I'm finished talking. So why do we still get sick if we have all these amazing, highly evolved immune systems? Well, the reason is the bugs are always one step ahead of us. I'm just going to give you an example here. This is Escherichia coli, E. coli. You've probably heard of this bug. It's a quite popular bug. It lives in your gut. So when you go for a poo and you have that lovely smell, a lot of that is the byproducts of the bacteria that live in your gut, including E. coli. I'm sorry to share that image with you, but it is a really prevalent bug in your gut. But sometimes it gets into the bladder and it's not supposed to be in there. It goes up through the urethra and it can initiate an infection. And we used to think infection was a really simple thing. We thought, oh, the bugs, just they look like little Tic Tacs here in this picture, and they sit on the surface of the bladder and they cause a bit of inflammation, and then you take some antibiotics and you're fine. That is actually not true at all. And if more recent studies have shown this is a lot more scary. What happens is the bugs actually dig into your bladder and hide inside it. So this is an image of a mouse bladder. It's quite sort of a smooth structure. And when you give E. coli into mice, you get this massive blisters forming. These blisters are actually full of bacteria. The bacteria have entered the cells of the bladder, gone inside and started forming these massive colonies, which are known as pods. And you can probably guess what happens to these pods. So the bacteria in there, when they're in there, they can't be treated with antibiotics and they can't be fought by the immune system. They're safe and they're hiding in there, and then they can just burst out like alien. And this is a video of some fluorescent bacteria bursting out of a pod. And this is a picture we took in my lab of some of these bacteria in the act of bursting out. So this is a bird's eye view of the bladder wall, and these guys are bursting into the wee. And there they are. They're really nasty and they're about to reinfect the bladder. So they go in, they hide, they regroup, they pop back out and cause another infection. So antibiotics can't really touch this, and also the immune system struggles to deal with these bacteria. Of course, there are defenses in the bladder. We'll hear more about this from Dr. Williams, but this is this purple thing is a big neutrophil that's trying to swallow some staphylococcus, and usually this works. But the E. coli that are in your bladder during an infection have an amazing adaptation to avoid neutrophils. So what you're looking at here are these little Tic Tac shapes. There's a normal E. coli, but if they've been in the bladder for a while and they burst out of those pods, they can burst out in a spaghetti shape, which is caused by a process called filamentation, which is actually not very well understood. So when they burst out, they look like spaghetti. And this spaghetti has several functions. The first function is it helps it stick to the bladder. Now, the bladder is constantly urinating, right? And that's washing away any bugs that get up there. And that's a defense mechanism we have as well. But this spaghetti stuff is very sticky, and it helps the bacteria to withstand like this fire hose of urine that's coming past. And the second thing is this filamentation confers an invisibility cloak onto the bacteria. And I'm going to play this video. This is very cool. The gray things are the Tic Tac shapes. E. coli that have the normal form. This is a spaghetti shape that's been false colored in green so you can watch it. The blue guys here are neutrophils, white blood cells that are trying to hoover up all the bacteria in the bladder. And let's see what happens. So munch, 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 munch. The neutrophils are hoovering up the bacteria, but they can't seem to either see or eat that spaghetti. I think they don't like the taste or they can't see it, and we don't really understand what's going on here, but it's great because if you can't be eaten by a neutrophil, you can carry on and reinfect and cause havoc. So that's another defense that these bacteria have. Here's another bacteria. I love this one. This is Listeria monocytogenes. And you can see what you're looking at is a human cell, and this cell is infected with Listeria, but Listeria is an intracellular pathogen, which means it only likes to live inside. And these black things are the bacteria. And why are they moving around, you might ask, Professor Rowan? Why are they moving around? They actually don't have much of a propulsion system themselves. What they've done is they've stolen the host propulsion system, in this case actin, and they make little comet tails of actin, and the actin shoots them around. And why are they shooting around a cell? Because they're trying to get to the next cell. There's another cell here. Can you see this? And these bacteria over here are desperately trying to get to the next cell. They're trying to spread, which is what bacteria want to do. But they don't want to go outside the cell because there's an immune system out there. So this adaptation allows them to spread from cell to cell without ever leaving the safety of that cell. And they're stealing our machinery to do it, which is really diabolical. I love this example. And I just have to say, there are about 1,000 examples of this kind of behavior that various bacteria employ to get around our defenses. I don't have time to talk about them all of them fascinating in their own ways. And this is why, back before the discovery of antibiotics, about 30% of all deaths were associated with an infection of some sort. a bacterial infection. And because there were no antibiotics, actually, we didn't even have antiseptics for surgery. People used to get surgeries and they just die because they got infected. So it was a really grim time to be alive until Alexander Fleming discovered penicillin. You've all heard this story. He left his petri dish out on the bench without the lid by accident, and a bit of mold landed in his bacteria and it caused a huge swathe of death. So there was no bacteria growing around this mold. And he thought, oh my goodness, this mold is secreting something that's killing bacteria. I wonder what that is. And the rest is history. It turned out to be penicillin, which is a very, very simple molecule shown here, penicillin G. It's a natural product of microbes. Antibiotics are not something that we designed as humans. It's something that the bacteria themselves have been using for billions of years to fight each other. And we just happen to stumble upon it. So antibiotics are an ancient and natural product of microbes. And they turned out to be a game changer. How penicillin works is really cool. So bacteria are covered by a cell wall. And this cell wall protects the bacteria from all of the environment. And if they don't have an intact cell wall, they burst open and die. So it's really important to have a good cell wall. And there's an enzyme called DD transpeptidase, I just call it DD for short, acts like mortar that keeps the bricks of this wall together. Penicillin simply flies in and blocks DD from doing its job. And when the cell tries to divide, it just bursts open. So it's a really effective drug against bacteria. And then it turns out that we discovered lots of other antibiotics in the last century. All of them were targeting some aspect of bacteria that we don't share. So any process that a bacteria does that we don't is a great target for a drug, because it means we can take the pills and it won't affect our own cells. It will only kill the bacteria. And we found lots of these, dozens and dozens of them. We had so many that in the 1980s, basically everybody said, yeah, we've got enough antibiotics. Let's just stop making them. Let's just stop discovering them. Let's just stop trying to make new ones. We've got plenty. Unfortunately, it turns out that the drugs that we have are starting to not work anymore. And I hope you've all heard about this because it's really super important. Alexander Fleming himself, when he was giving his Nobel lecture speech in 1945, said, guys, I just want to warn you that it looks like the antibiotics don't always work forever. If you use them for a long time, the bacteria learn how to get around them. And I'm really worried about this. And he said things like, I'm really worried that people will take antibiotics when they have a cold, and they shouldn't. They should only take it for bacterial infections. Nobody listened to him. And for decades and decades and decades and generations, people worried about antibiotic resistance, but nobody did anything about it until we've got to the point where antibiotic resistance is rife. And what do I mean by that? I mean that the drugs don't work because the bacteria can fight them. In this case, bacteria can learn to produce an enzyme called beta-lactamase, which basically acts like scissors and just pops open the penicillin, and it can no longer work. So if you've got the beta-lactamase gene, it doesn't matter how much penicillin you are exposed to, you just snip it in half and you're fine. And these beta-lactamase genes and other genes like it can be passed around like business cards between bacteria on little things called plasmids. So once one bacteria has the business card, it can just pass it to all his friends. Before you know it, everybody's resistant. So if you have a petri dish with lots of white bacteria that are susceptible to antibiotics and one mutant that has a plasmid that makes it resistant, what's going to happen if you put penicillin on this plate? All of the white ones are going to die and all the red ones are going to grow. You're going to select for the resistant strain. This happens in a petri dish, but it also happens in a hospital ward where everybody's being treated with an antibiotic. It can happen on an airplane flying from Hong Kong to Singapore. It can spread really easily once it's out there. And that's why resistance is on the rise. And it's on the rise mostly because we're abusing and misusing antibiotics at a terrible rate. So we take antibiotics when we have a viral infection. It won't work against viruses. People can buy antibiotics online for 30p at Canadian pharmacies. And people are always taking antibiotics when they don't need it. They're not finishing their course of antibiotics, which means that the bacteria aren't killed and then can become resistant. We're dousing our agriculture, our viticulture, our aquaculture with antibiotics that aren't needed. And it's in the environment, just like that petri dish. It's seeped into the environment. It's teaching all the bacteria to resist all the antibiotics. And it's a perfect storm now. We had 20 antibiotic classes discovered in the golden era between 25 and 87. Resistance to all of them is on the rise. And there's been a discovery void ever since. The number of new antibiotics just keeps going down and down and down. There's been a few recent successes, but we're still pretty much at the end of the road for many antibiotics. This means, what does this mean? This means one of you could cut yourself on a thorn or scratch yourself. You could end up with an infection that cannot be treated by any known antibiotic, and you would die. And this is amazing to think you could scratch your finger and die, but this is what used to happen before Alexander Fleming discovered antibiotics. And that's not some scary future. That's right now. Right now, almost 5 million people across the globe die with an infection that's associated with a resistant bug, and they can't be treated any other way, and they die. And this is unbelievable. It's kind of, we're sleepwalking into an apocalypse. And I could talk on and on about, I love this topic. It's really important to me personally. We all need to take action, whether that's just not taking antibiotics ourselves when we don't need them, or joining the Applied Medical Sciences program and becoming a scientist and discovering alternatives to antibiotics. There's some really interesting things on the horizon. Probiotics, so good bugs, can be used to fight bacteria. We've got some vaccines against, of course, vaccines against bacteria are quite useful. People are using viruses to fight bacteria and developing chemicals that can block bacteria from binding to your cells. There's all sorts of cool strategies, but we don't have enough of them. And we need people like you to come and join the fight. So I really hope that you'll consider going into biomedical research and helping with this problem. And now we're going to hear from Dr. Williams, who's going to talk about the other side of the coin, which is, drugs are great, but we really need the body to step in and help too. So over to you, Dr. Williams.
Thank you very much, Professor Rowan. So I'm going to talk a little bit about our immune system. So this little figure on the left here. So this is our family of immune cells. It's not the complete family tree, but it's probably the most important ones, the ones we know most about. Professor Rowan already talked about neutrophils in the middle there. So I'm going to talk a little bit about macrophages. So top row, second from left. And B cells, so down the bottom, because B cells are really important because there are immune cells that make all our antibodies. Right, so next slide. So why do we have an immune system in the first place? So, well, this is a person sneezing. So a sneeze can probably travel, I don't know, 10 meters, can travel at 100 miles an hour. Stay in the air for 10 minutes or so. And each of those little particles you see could potentially be loaded with a whole bunch of different viruses or a whole bunch of different bacteria. So we've evolved this immune system. We don't get sick every day, so our immune system must be pretty good at generally protecting us pretty much most of the time against all these potential pathogens and nasty microbes that are in the environment. So this little video here, so let's start with our nasty little bacteria again. So this is a type of white blood cell, a little bit like a neutrophil, called a macrophage. So what this macrophage is doing, it's sensing its environment. It can detect that there's a bacteria around. It chases after the bacteria, latches onto it, and starts undergoing this process of engulfment. It's what we call phagocytosis. So pretty much these macrophages hoover up, or at least try to hoover up, all these bacteria that enter our body, whether that's in our lungs, in a wound in our skin. So these bacteria, these macrophages are really, really important for providing us with that first line of defense against invading pathogens. And they do this, so they hoover up the bacteria and they engulf it into something called a phagosome. And this is an intracellular vesicle which contains all sorts of digestive enzymes and reactive oxygen species that's able to degrade the bacteria. So they're a really good first line of defense. Right, we go on to the next slide. So I'm going to talk a little bit about B cells as well. So one of the other things that macrophages do, they can activate other cells of the immune system. One of these other cells that they activate are our B cells. So once B cells become activated, they start proliferating and they start differentiating into these antibody producing factories that we call plasma cells. And each one of these plasma cells is probably able to synthesize 1,000 antibody molecules per second. So they're really, really important immune cells that synthesize all our antibodies for us. Next slide, please. And antibodies are important because we know that they are the little molecules that attach to the surface of pathogens. So we have antibodies that we make that are specific to bacteria, and we have antibodies that we make that are specific to viruses. And each one of these antibodies is specific. So we need a different sort of antibody that can recognize a bacteria and a different sort of antibody that can recognize a virus. But what these are really good at doing, they coat the surface of these viruses and bacteria. And that enhances the way in which macrophages are able to engulf and phagocytose and degrade these pathogens. So it's linking these two kind of immune cells together with two different types of mechanisms. And they can also coat the surface of viruses and stop them from infecting cells. So if you go to the next slide. And you probably have heard enough about this virus by now, SARS-CoV-2. It's the coronavirus which causes COVID-19. And we know that this virus latches onto our own cells through this little protein on its surface called the spike protein. And the receptor on our cells, so the cells that line our respiratory tract, is known as ACE2. When a virus latches on via its spike protein to ACE2, it can then invade our cells lining our respiratory tract and lungs and can cause pneumonia and all sorts of things. This is where antibodies come in. So antibodies are really good at attaching to this spike protein. And if we have enough of them that attach to the spike protein, they can stop that virus from interacting with that receptor. So essentially it neutralizes that virus. It stops it from infecting our cells. So antibodies, really, really important. And scientists, if you have the next slide, please, scientists can take advantage of this natural production of this antibody. And this is why we have vaccines. So what vaccines do, well, most vaccines do, is that they're really good at inducing an antibody response. So you might be familiar with this. This is the Pfizer BioNTech COVID-19 vaccine. And it's a little bit funky because instead of a protein or a killed virus, what this particular vaccine does is it uses a little piece of genetic material known as messenger RNA. And we know that messenger RNA is really good or is essential for making protein in the body. So messenger RNA is translated into all our different proteins. So what these scientists have done is that they developed this vaccine that contains the messenger RNA that codes for the spike protein of SARS-CoV-2, of COVID-19. So that when we get vaccinated with this mRNA protein, we make lots and lots of antibodies that are specific to that spike protein. Actually, interestingly, in the last week, the Nobel Prize for Physiology and Medicine was awarded to the people that discovered this mRNA vaccine technology. So really, really important technology for protecting us against disease. And we do lots of COVID-19 research, but we do lots of research at UCL. I just thought I'd highlight some of the really important contributions that scientists at UCL have made towards COVID-19, not just in basic science, but also in the social sciences, how it's infected young children, developing a CPAP device so that patients can breathe more easily when they have severe COVID-19, and understanding the mechanisms of how this virus infects us and how our immune system responds to SARS-CoV-2. Right, I think I will end it there. Give us plenty of time for questions.
I love your videos.
Funky videos.
I might have to borrow those for some of my lectures. Great. So we got some questions, I hope.
So unfortunately, for now, we've received no questions.
Oh, that's okay. Maybe we can warm everybody up. I think, I think it's really useful for students to hear from current students. And I thought maybe, Erin, you could kick off things by just maybe saying why did you choose this university and this course?
I mean, firstly, I know a lot of students like to get confused with, the classic biomedical sciences course. But when I was researching, especially firstly the course, I discovered that the variety this course offers you with its optional modules and everything like that, it's a very tailored course to you. It's almost like you get to build your own course. Like, when we have those Build-A-Bears and all those shops like that, it's almost like you're building your own course right here. And of course, UCL, everyone's heard of it. It's a top uni in the world, not just UK. As well as that, researching more about UCL and seeing how further we are in research in terms of exposure to research scientists, labs, and everything like that. It was a very convincing university to choose, despite everything as well. So I think that both of those parts played a big role in specifically picking applied medical sciences. And I mean, as well as that, with this course, sometimes we're based at the Royal Free Hospital, which again is a new environment, typically different to what a average university environment is. Or sometimes we're based at our Bloomsbury campus. So it gives you a wide range of perspectives. And even our professors are lead researchers, doctors, or they're still, conducting research. So we really have that opportunity to network and talk to a lot of people regarding what we're passionate about or what we want to delve deeper into. It's not like it's tied down to one specific area. So I guess that did kick things off because we've come up with quite a few questions. So one of the questions, the first basic question is, do we get a clinical placement in this course?
I guess not specifically that, but in the year 3, there's a research project, and some of our students do go to work with clinical researchers, usually in the lab or maybe working with patient data. So no, we don't get a clinical placement, but we do get some of our lab practicals are geared towards clinical stuff. Like we have the practical on, Erin, you're probably a bit more familiar with than I am, but there's stethoscopes and spirometers.
Yeah.
We do all sorts of stuff with clinical measurements for physiology in the laboratory. And we have research projects that are related. Some of them are related to clinical scenarios. Does that answer the question?
Yeah, I think it's it's worth reminding everyone. It's not a medical degree. It's it's not a medical qualification. It's a Bachelor of Sciences degree. It just that it's quite clinically orientated with the subject material that we cover.
Perfect. I think that should answer their question. And this also, again, this links with another question that someone asked is, how does applied medical sciences differ from biomedicine?
We get this question every time. Do you want me to answer that? I think you've already covered this, but essentially, biomedical sciences. So the UCL Biomedical Sciences course is great. It's a really prestigious course. You learn lots and lots about science, biology, biological science. In our course, you learn both science and medicine and how they link together. So our course is a lot more medically oriented. We have a lot of lecturers who are active clinicians who'll be doing case reports and taking you through patient scenarios. Basically, it's a mix of both, whereas biomedical sciences are much, much more geared towards the basic science.
I suppose biomedicine as well, and biomedical sciences. It's almost a training degree to become a biomedical scientist. So our degree, I think our students go and do a much broader range of different things after they graduate.
Perfect. I think that answers that question. So now I guess this question would be to all of us. What is the best part of applied medical sciences?
The best part? Why don't you answer that? Because you're a student, you can tell us.
I mean, the best part of applied medical sciences, I guess it's, as you've already explained, it's more like bridging a gap between medicine and biomedicine. It's quite a lot of theory-based, but we also have quite a lot of practicals we do, such as measuring our ECG or things like that. I think it's the more in-depth that we go that makes applied medical sciences personally my favorite part in it. And I personally like the practicals a lot more, just because I know currently a lot of these students would be doing their A level. So when you get to university, you realize that practicals are so much more fun in that sense, that you can go ahead and figure out and research different things. So now our next question is, so this I presume is something from the presentation. On the image of the E. coli, what was the string around the cell?
I think you're talking about that scanning electron microscopy image where there were orange bacteria and they had all this yellow stringy stuff around them. What those are they're called flagella, and they're the appendages that allow the bacteria to move. And these bacteria have to go a long way. They start in the gut from your back passage, and they crawl across your skin and up into the urethra, into the bladder, against the flow of urine. It's really remarkable. So these little appendages help them swim, and eventually they help them to attach. They help them to dock onto the bladder to initiate the infection.
Interesting. Now, another question similarly. So it says that we said that the coronavirus vaccine contained mRNA that codes for the antibodies that attack the virus. But why doesn't the vaccine just contain the inactive pathogens or antigens so that the body's immune system can produce the specific antibodies itself, like a traditional vaccine?
Yeah, excellent, very good question. I like this question a lot. So there are COVID-19 vaccines available that do that in the traditional vaccine sense. So you might have heard of the AstraZeneca vaccine. So that uses a viral vector that contains the protein, or that at least makes the protein, the spike protein of the virus. The beauty of an mRNA vaccine is that, one, it's cheaper and it's quicker to manufacture. We also know that SARS-CoV-2 is evolving rapidly. So it's evolving ways in which it can escape our antibody responses, for example. The beauty of an mRNA molecule is that you can easily alter it. And you can easily alter it so that it makes the spike protein of the most recent variants of the virus that's in the human population. So it's both about manufacturing is a lot easier. It's much more flexible in the way that we can alter that vaccine. And also, when an mRNA molecule is in our own cells, it makes lots and lots and lots of spike protein. So it's a good way of extending the lifespan of that protein that you want your immune system to respond to. So there's lots of reasons. Both approaches have been used. So.
Now, our next question brings us back to the whole application process. Is the first part of it is classic. What are the entry requirements for the BSc Applied Medical Sciences course?
Yeah, so I've just tried to put in the chat. There's a on the web. You can look them all up. Basically, you can look up if you're an A level or if you have the IB, or if you have other qualifications from a foreign country, for example. This website's really good. You can just look up your specific situation. But for classic A levels, we're asking for 3 A's. You have to have an A. One of those A's has to be biology, and you have to have +1 from chemistry, maths, or physics. So you must have an A grade in biology and 2 other A's, one of which has to be chem, math, or physics. IB, I believe it's
37, is it?
That's a contextual offer. Yeah, 38 now. It's gone up to 38. Yeah. And then obviously we have contextual offers. So if you have particular situations, you can look up your situations and see what the dropped offers are there. But yeah, it's all on the website. Did that enter the chat? I tried to type it in.
I think it has entered the chat, yes.
Great. So just have a look on that link. And if you have any questions about anything, you can email me. I'm the admissions tutor. But if it's really technical questions about applying, you're probably better off emailing the admissions office, who really know all the fine details about that.
I mean, since you're an admissions tutor, this question is mostly for you. Should you make your personal statement for Applied Medical Sciences very sciencey?
Sciencey. Well, yeah, I mean, that would help. The personal statements are important because we get way more people applying than we can take. So you do need to put some thought into it. I think one of the things, it's important to make sure that your statement is relevant to this to this particular course, you have to show us why you want to be in this course, why you're a good person. But really, don't worry about sort of being sciencey. What you want to show is that you have passion and curiosity, and you have the drive and then the ambition to want to be on this course. And obviously, it's important to show that you're a well-rounded person. So you can mention if you have hobbies, or if you've done volunteering. Just don't worry too much about it. But please don't get someone else to write this for you. I mean, it's really obvious when a personal statement's been sort of manufactured or bought. Make it from the heart, and it doesn't have to be perfectly written. It doesn't have to be written in perfect English. We know that a lot of our students come from other countries. When it's not an English exam, we just want to see the authentic self, your passion and your interest. So yeah, just put some thought into it, put some passion into it, and you'll be fine.
Perfect. Just adding on to that just a tiny bit more. They've asking what kind of opportunities should they be looking for, i.e. work experience or essay competitions or something along those lines? What would be the most useful for applications?
I have to say, honestly, we don't necessarily look at things like that. I know that if you're applying to medical school, these kind of things are really important. It's okay. I mean, just tell us what you've done. You must have done a few things that are interesting. You can mention those, but I don't think that's going to make or break your application. We're looking for people with the good marks who have a really nice, authentic letter. Don't worry about, having to, volunteer for 10 million hours at a hospital. It's okay. I mean, it's good for you to do stuff like that, but it's not going to make or break your app, I don't think.
Perfect. So now the next two questions are a bit more medicine based. So firstly, are genetics and virology both studied in the medicine course at UCL?
So we can't answer questions for the medicine course. Do you mean applied medical sciences?
Let's assume that's what they mean and answer it that way.
So, yes. So you do a little bit of both in year one. In year two, we have a human genetics course, and as part of the flexible nature of our degree program, you can take optional modules in the subjects that you're interested in, and of course UCL, one of our sister departments, provides lots of virology and the immune response to viruses type modules. So certainly there is lots of opportunity to study virology at UCL. Absolutely.
I think that goes ahead and answered this question. Now, of course, there's one more question. Can you practice medicine with an applied medical sciences degree?
Nope. It's not a medical degree. You need to do an MBBS or equivalent medical degree.
But as you mentioned, a lot of our students go on after graduating to go into medical school. And actually, some of them say they're really bored in their first year because they learned a lot of it in our course. And so they had to sit through a whole year of what they already knew, which is good. It means you're well prepared. It is quite a large fraction of our students who go on to do that. So, yeah.
Perfect. So now the next question is, could someone survive with a very limited amount of antibodies in them? So I assume that means as in the immune system as a whole is a bit weakened.
Yeah, I'd probably say a lot weakened. Antibodies are really, really important. So there's some people that have inherited immunodeficiencies, and they will receive intravenously infused antibodies to help protect them against viruses and bacteria. Some people who are on cancer immunotherapy also have a reduced antibody response, and that makes them have an increased susceptibility to infections. So I think you could survive, but it would make you very susceptible to succumbing to an infection.
I mean, it's worth noting that the older you get, the less well your antibodies work, and so aging is also associated with having increased infections, because basically everything's your T cells, your B cells, everything's sort of bits are falling off. Speaking as an oldie, I'm starting to worry about that.
So now our next question is, how can a deeper understanding of the interactions between pathogens and the host's immune system at the molecular level lead to further development of a more effective vaccine or treatments for infectious diseases?
What a great question. Sorry, that's an amazing question. I mean, that is exactly what people are trying to do. We're trying to harness, we want you to apply to our course, whoever that is. Divya. Please do apply, because these are the kind of questions that we want to answer as scientists. And this is like the final frontier, I think, is harnessing our own immune systems instead of just using drugs that are toxic and nasty, trying to find ways to harness our own immune system. Maybe Dr. Williams probably has something to add to this, but yes, please come here and study that. What do you say?
Well, no, it's kind of you're answering it in your question, really. Yeah, absolutely. I mean, that's the approach that people are using now to try and figure out how we can design better vaccines, trying to figure out what the interaction between the virus and the host is, and can we interfere with that interaction. So it's not just infectious diseases, it's cancer as well. Can we better understand the molecular interactions that are driving cancer and what's preventing our immune systems from clearing that cancer? And can we design novel therapeutics that take advantage of those new discoveries? Can we interfere with those mechanisms that cause disease? So yeah, good question. Well done, Divya. Good question.
So another question we have is, wouldn't it be hard to promote antibiotic alternatives or to stop people from overusing them in poorer countries if antibiotics are seen as the cheapest option? What theoretically could we do to tackle this situation?
That's a huge question. I was just sitting on a panel from a bunch of people who came over from India who wanted to exchange ideas about how to, because India has a high level of people just taking antibiotics for no reason. The pharmacies, you don't need a degree to become a pharmacist, and it's a really popular drug. Yeah, I mean, the only answer here is education, right? You need to teach people, why would you want to take a nasty drug that's going to kill your own friendly bacteria if you don't have a bacterial infection and you've got a cold? But it's a huge, huge problem, and you need not just science, you need behavioral science, you need to sort of economical sciences, like all of the disciplines come into this question. The whole problem with antibiotics is a multidisciplinary nightmare. And yeah, it's a real problem. And in fact, if you want to come to UCL and start thinking about how you might solve this problem, that would be great, because we need lots of bright ideas.
Perfect then. So the next question is it possible for the mRNA to mutate in the future? So I think this is a part 2 of the previous question about mRNA that we had.
So you mean the viral RNA? So I'm taking it you mean the viral RNA.
Or maybe the vaccine.
The vaccine won't mutate because it would require it to be engineered to incorporate new mRNA sequences. But certainly the mRNA in the actual SARS-CoV-2 virus, that is mutating constantly. So we've got a really high mutation rate. That's why we keep getting mini outbreaks right across the globe of new SARS-CoV-2 variants. Omicron B1.42, Delta this, Alpha that. So yeah, it's constantly mutating.
So now our next question is, how do autoimmune diseases actually arise? What is causing our immune system to go ahead and attack our own body?
Right. Okay. So not everyone gets autoimmune disease. So the first part is probably people that do have some sort of genetic predisposition to developing autoimmunity. There's a thing called normally our immune system is programmed to not attack our own body, and sometimes that programming can break down, particularly in later life, when the immune system's not functioning as it should. So there are ways in which the immune system, the programming of the immune system, can break down as well. It is complex. So it's a combination of genetic and environmental factors, probably.
Perfect. And just a bit not sidetrack, but a bit further on. The students have asked, can we explain to them how bacteria jump from animals to humans?
Well, I keep chickens, right? Believe it or not, I have a flock of chickens in my back garden. And if they've got bird flu, if they've got bacteria, and I handle the chickens or the eggs, and I don't wash my hands, I can easily. I mean, bacteria are but usually viruses are very good at jumping around. And in fact, SARS-CoV-2 probably jumped off of an animal into humans, and The reason why that's so devastating is because when a bacteria or a virus jumps to a new host, the host isn't used to them and has never seen it before. And that's why it's just going to rip through the body and cause all these awful pandemics. So yeah, animal to human transmission is a big problem. And it's simply down to hygiene. It's either through eating, not washing your hands, or sometimes people eat bush meat that's infected, and there's all sorts of ways that you can get infected through animals. So yeah, hygiene is really important.
I think it's an issue with climate change as well, where we have a reduction in biodiversity and a reduction in biomass that's bringing humans into closer and closer contact with these animal species that we shouldn't really be in contact with, and they can harbour viruses and bacteria that we've never experienced before.
So now our next question is kind of, I guess, a cool, sci-fi question. Do you think we would ever be able to develop an antibody that can somehow automatically always change shape to fit whatever antigen it encounters? So almost like a, shape-shifting one.
We already have that. Anyway, go ahead, Dr. Williams. That's you just kind of describe what really happens. But.
Yeah, I mean, not a single molecule, but a single molecule is fixed in its structure, and it doesn't really adapt over time. But the B cell population that we have can adapt over time, and the B cell population can change in ways that will adapt to what viruses are around humans at any given time. I'm not sure about the other sci-fi bit about it that we can develop a nano antibody that can change shape.
I don't think you need it. I mean, the human body has already come up with this amazing, there's billions of combinations that the antibodies can take depending on what they see. And I think we already have, it's almost like science. I mean, I think it's almost as amazing as science fiction, how it works right now. But yeah.
Our next question, a bit more about antibiotics. Do antibiotics work for those bacteria, and they give an example of Listeria monocytogenes, that can invade cells and use the cell's machinery like viruses? If not, then what are the treatments available for these special type of bacteria?
That's a really great question. So yeah, there's a lot of bacteria. There are a lot of intracellular bacteria, not just Listeria, but the Staphylococcus, E. coli. They can invade and take up residence inside cells. But there are special antibiotics designed for intracellular infection. Chlamydia is a great example. It's a obligate intracellular parasite, but there are drugs that will target, like azithromycin, for example, because it can get through the membrane of the cell. There are other antibiotics that cannot get through the cell membrane. So you have to use the right kind of antibiotic for those particular bugs, but they do exist. Although all antibiotics, as I mentioned, are getting less and less useful. So,
I mean, one of the, I guess, final few questions we have is, I'm a year 12 student. Where or how can I find work experience in order to learn more about biomedical sciences? So what would be an ideal place where they can learn more about biomed?
That's a tough question. Did you do any work experience yourself before you applied?
Honestly speaking, no. The only thing that I did extra was the extended project qualification, the EPQ, and that I did on embryonic stem cell therapy.
Oh, wow.
But I felt like the EPQ, it kind of gave me some skills that I use in our course today, because we had, you have to go ahead, research, referencing, everything as such. So I feel like, not exactly, I haven't done any work experience, but I definitely did something that gave me the skills that helped me in this course.
On applications, I sometimes see people have volunteered at their local GP surgery, shadowing a GP. I've seen people saying that they've managed to find a laboratory that will allow them to work for a couple of weeks, shadowing or even working. So it is possible. You really have to be proactive and try to find these opportunities. I don't know if they're advertised. I think it's usually just a student saying, I really want to do something, and they just are really tenacious and they go ask lots of questions and send lots of emails. It's not easy, but students do manage to do it.
Yeah. Perfect. So our next question is it true that in order for bacteria to better resist bacteriophages, they lose antibiotic resistance? thus meaning a combination of phages and bacteria could be used for more effective treatment?
I don't know if that is true. They're two separate processes. So antibiotics and phages do things differently. But it is true that phages, as I mentioned, are now being considered as an alternative therapy. In fact, it's interestingly, they've been used for decades in the former Soviet Socialist Republic, like Georgia, for example, that you can get phage therapy there. And it's becoming increasingly popular here, and we're taking a second look at it now that we're running out of antibiotics. So phages are interesting.
Perfect. And one of the final few questions that we'll take is, how can UCL researchers use new methods and tools to find and target specific parts of harmful germs for better tests and treatment, especially when dealing with a new type of infectious disease?
Wow, that's a big question. That it would be a multi-pronged approach using lots of different techniques, both biochemical, molecular, cellular techniques, genetic techniques. One approach would be to take a whole bunch of different antibodies of all different specificities and see which ones stick to the virus. You can then narrow that down molecularly to see where that antibody sticks to at a molecular level, at an amino acid level, and then work out what those amino acids are doing. What are they doing? What is the virus using them for? You can then develop drugs that target that tiny little aspect of that viral protein to see if you can interfere with its function. Crystallography studies, biochemical studies.
Yeah. Nobody at UCL studied coronavirus until the SARS pandemic, but almost instantly, people around the campus started to, they had respiratory cell cultures. So people had mini lungs, and they started to, because a new virus, right? You don't know how it works. So people immediately pivoted to that. So scientists are quite good at pivoting to new diseases. And there's, as Dr. Williams says, there's loads of different ways.
Even so far as now using artificial intelligence to help predict where these, new targets might be.
That's really exciting as well.
Perfect. So in terms of questions, I think those were about it that we have. So if anyone has any more questions, feel free to email admissions office or Professor Warren or even Dr. Williams. And once again, I firstly thank the Professor Warren and Dr. Williams for joining us today and giving everyone a taster session of how it's like to study at UCL, particularly the Applied Medical Sciences course. And thank you to everyone that joined us here today. And we know, sometimes you might think of questions after the session. So feel free to either check the website or the prospectus, or feel free to always email admissions office or any one of us to get your questions answered. As well as this, please could you fill out a survey that we'll be sending out, just so we can make this better for next year, and any improvements or any comments you may have on that. Apart from that, I think, and also for those who are wondering, this lecture is recorded and everyone will receive a link. So don't worry if you feel like everything's a bit too much to process in one go. You will be sent a link. And apart from that, I think that is good evening for me.
Yep. Thanks very much for coming.
Goodbye from me. Thank you all.
Thank you all for coming.